While you're getting set up, I'll introduce Professor Chang. Our next speaker is Professor Michelle Chang, the A. Barton Hepburn Professor of Chemistry at Princeton University. She received her PhD at MIT under Joanne Stubbe, where she provided direct evidence for radical transfer in ribonucleotide reductases. She then carried out postdoctoral work with Jay Kiesling at UC Berkeley, pioneering microbial engineering approaches to produce complex natural products. Her own laboratory has become internationally recognized for discovering and engineering enzymes that catalyze reactions difficult or impossible by traditional chemistry, such as selective CH functionalization, halogenation, even biological fluorination. She has also advanced microbial biosynthesis of terpenoids, fuels, and other valuable molecules, bridging enzymology, chemical biology, and synthetic biology. Professor Chang has been honored with awards, including the NIH New Innovator Award, the NSF Career Award, and the Royal Society of Chemistry Centenary Prize. Today, she will speak on the discovery and engineering of new enzymes for biocatalysis, highlighting how nature's catalysts can be repurposed for chemistry and biotechnology.
Okay, well, thank you, Romus, for that kind introduction. Also, thank you to you and Claudia for the invitation, and then our visitors from Japan. So my lab works, as Romus mentioned, in biocatalysis. What we are interested is in taking advantage of the chemistry we find in nature, just like everyone else that you've heard speak from today, and ask how we can use the catalysts or enzymes to do new chemistry. So I think most of you are well aware of the role of enzymes in industry, not only for scalable products like food production, but also things like the synthesis of pharmaceuticals. And so Merck is a leader in this area and is shown, for example, in this case, with this transaminase, that by replacing a chemical step with a single enantioselective enzyme, they're really able to reduce the amount of steps, increase the yields, reduce waste, as well as reduce the cost. And that's really highlights the quantitative value that enzymes bring. And so there's many classes of enzymes that are used very prevalently already, such as making mostly oxygen and nitrogen containing chiral building blocks. And these chemistries are well understood and well used. But as Hui-Min pointed out, there's still a lot of chemistry out there that we need to construct molecules the way a chemist does. And in order to do so, we need to tap into things like carbon-carbon bond forming reactions, other types of reactions where we can build complexity and not simply carry out functional group conversions. And so that's what I'll talk about a little bit about today, is our work in looking at enzymatic CH activation, and then also the study of mechanism, how that works in our system, how we use that to engineer new reactions, and I'll tell you a little bit about enzyme discovery. So my lab has become interested in amino acid chemistry at different length scales. So if we start from the small n, non-canonical amino acids, the making of amino acid-based natural products such as alkaloids, peptides and peptide drugs are very important, obviously functionalized proteins. And if we understand the chemistry of amino acids, how we extend that to peptides, and that allows us to think about, again, making bioorthogonal tags for protein modification. So this started actually, as everyone else has mentioned today, from a set of papers from a Japanese group, the Iwadare group, who reported the biosynthesis of these two non-canonical amino acids in Streptomyces cattleya, a soil microbe. And so my lab has focused on both of these, but more recently have been more looking at these terminal alkyne amino acids for their use as click handles. And this work is published, but this is kind of what led to this research direction for us. It's actually quite common that non-canonical amino acids are derived from amino acids. So in this case, you can see starting from lysine, we do halogenation, that's site selective and regioselective and stereoselective to replace that hydrogen with a halogen, which I'll get into later. You have an oxidative cleavage reaction and then formation of a terminal alkyne through a PLP-dependent step. And so what we really got focused on is this first step, as I mentioned, because it's a CH activation, which is an important chemical transformation. Basically, every molecule has H's on it, but how do you choose a specific H to introduce a group that gives you a chance to do downstream chemistry selectively? And halogenation and other sorts of things that you might use in place of halogenation could be quite interesting in that regard. So this is a radical halogenase. So it uses a radical mechanism to abstract the CH and replace it with an anion. And so what's very interesting about this reaction is there's a lot of enzymes that do CH activation. They do them usually through the use of oxygen, except in the cases where Hui-Min is talking about new to nature reactions where you use a photobiocatalyst, but these also generate radicals that are reactive. But you generate this high-valent iron 4 oxo species that abstracts a hydrogen from the substrate. And most enzymes that do CH activation will place the oxygen back on the molecule. We call that rebound. Rearrangements can occur. You can get different transformations, but typically the oxygen will go back in. But what halogenases do instead is they use a bound anion to instead to replace that OH, and it carries out an alternative rebound pathway where instead of rebound with the closest atom here, which is the kinetically and thermodynamically preferred product, it creates an open coordination site to replace a protein aspartate with a protein-bound anion. And so if we can understand that better, then maybe we can understand how you engineer the transfer of a variety of different anions. So the way my lab took to start understanding this question is using protein engineering, is that halogenases are actually related to their hydroxylase counterpart. So these are highly related, which I'll get into later. And to distinguish them can be like looking for a needle in a haystack. And so, but what you see here is when you have a specific family, they do break down into phylogenetic relationships where these are maybe about 30 to 40% similar to the halogenases. But that means if you look in amino acid space, you're talking about things that might differ by 200 amino acids or so. So how do you identify the specific molecular basis for this halogenation reactivity? And that's what we're interested in. So we took this one relatively more related hydroxylase. What Monica did in the lab was to cut them up and basically shuffle the halogenase and the hydroxylase together and ask which parts of that halogenase would confer halogenation activity and what was the minimal number of amino acid changes you could do that. In order to do so, she developed this high throughput assay where you could put the enzymes in to make this terminal icon amino acid. If it chlorinates, you can make this. You can click to it. Those are fluorescent. But if it doesn't chlorinate, then you get no click reaction, and then those are non-fluorescent cells. And so she was able to figure out that there was only 14 residues that conveyed halogenation activity. I'll describe the most important two developed in this step. The rest are involved in chloride binding and other things associated with that. And so what we decided to do is to look at these mechanistically. I'm not going to go over all the details of the reaction because it's archived and it will be published soon. But what we started to do is to look at crystallography to explore some of the early steps in the reaction pathway because these were not characterized in halogenases. For example, this is the first case of seeing the initial activation of oxygen to have a structure of it in an alpha-ketoglutarate enzyme. There's been some question whether it does the same, whether it makes this intermediate or not. We could also see when we use vanadium oxo as a stand-in for the iron oxo because it's not reactive, so it represents a structural model of the intermediate. What Eli found was something interesting is that he couldn't place the oxygen and the chloride ligands. Instead, he saw a combination of isomers. And what that meant is that the oxo was either expectedly close to the substrate or inline, or it would be distal in the equatorial plane and offline that would be further than expected. The interesting thing was this was studied by the Solomon lab, who had carried out a computational and spectroscopic study of a related halogenase CRB, and mentioned that there were these two different pathways. In their case, the issue here is if you do the expected CH activation through this inline pathway where these are adjacent to each other, that's rapid, but then rebound is rapid and you get hydroxylation. So what they proposed was that radical abstraction took place from this distal orientation through a pi symmetry pathway, and that placed these farther enough apart that then rebound with chloride would be imminent. So just to confirm, we use spectroscopy just to also support the existence of these two isomers because crystallography can be a little bit challenging that way. We also measured the barrier to this offline versus inline H-atom abstraction using computation. So you can see when it's close, the energy barrier is much smaller than when it's far. We can actually measure the rate of H-atom abstraction using stop flow. And what we see is that our expected barrier is consistent with this one here. And so then that raises a question of, if it's adjacent, why does the chloride transfer in place of the hydroxy? And so what Yanni did in the lab was carry out a DFT calculation and showed unexpectedly that the hydroxyl migration or the isomerization of the chloride and the hydroxyl group had an unexpectedly low barrier that actually competes with rebound. And what he found was the two residues that we had found through our Monica's shuffling experiment were key to that in terms of making interactions with the succinate to drive it to a 5-coordinate species, as well as hydrogen bond interactions with the hydroxyl group. And then this is further supported by crystallography to show that a key water in that process from the DFT is lost when you mutate one of those key residues. And so that gives us a new model for thinking about halogenation. In this case, we believe that this oxygen binds in the empty coordination site as expected. We have a crystal structure of this intermediate to show that the oxygen attacks the alpha-ketoglutarate, so a cofactor which leads to formation of the oxo. The H-atom abstraction takes place next to the substrate in an inline position, and what we know now is these can exchange position based on this asparagine 224, and that allows halogenation selectivity. And so that gives us sort of a new mechanism to think about the mechanism of anion transfer and a new set of knobs to tune in that reaction. So I'm just going to switch our attention to just thinking about what engineering. So I didn't mention this before, but halogenase families tend to be have very different be very different in primary sequence space. Each individual family tends to take a specific type of substrate. So ours takes, as I mentioned before, lysine, as well as these aliphatic amino acids. These tend to be reserved for sp3 hybridized substrates, so they tend to work on these methylene groups. So we had been hoping that these would have more diverse substrates, but it is what it is. But it allows us to look at different aspects of substrate selectivity in great detail. For example, Eli has looked on why you get 4 versus 5 halogenation, why you can get ornithine versus lysine in terms of chain length. And I'll just tell you a little bit about example of what Yanni's done for looking at mono versus dichlorination. So I just told you that halogenation is very distance dependent. Actually, we believe it has a sensitivity of 0.5 angstrom or below. So how do you actually halogenate on two sides of the same substrate? They're in very different orientations. And so using that same approach where, again, the dihalogenase and the monohalogenase are too far apart in sequence space, what we can do is we can construct a model where we map on one set of residues onto another sequence. And in doing so, then what we can do is to identify basically the key residues, which are these 4 right here, that really control selectivity. And in doing so, we can engineer things from monohalogenases to di- and trihalogenases. And that gives us the positions that accommodate movement of the substrate, but still continue to halogenate instead of converting to hydroxylases. And so using that information, Gabby can do things like engineer azidation. So these enzymes with, again, simple mutations can quantitatively convert lysine to azitolysine, even in the presence of competing chloride. We can use these because amino acids, as I mentioned before, are building blocks to make various different types of molecules. For example, we can make these different lysines. We can put them into peptides by in vitro translation. And we can use those to do both with hydroxylysine as well as azitolysine, various types of downstream chemistries. For example, obviously the click reaction, we can also reduce these to the diamines and do things like put them into polymers. And so, as I had mentioned a little bit, different length scales, how do we expand the scope of biological halogenation? As I mentioned before, it's very distance specific. And so in terms of geometry of the active site, so to convert something into that recognizes an amino acid to something that recognizes a peptide sequence can be quite difficult. So our approach is to take something that catalyzes a different reaction on a peptide and ask whether we can do something like direct protein azidation. So in this case, we can use a method pioneered by Alice Ting in terms of proximity labeling, where we display the substrate and the enzyme on the surface of yeast, and then through another fluorescent click reaction, then we can sort through these things by fluorescent cell sorting. And so this is just, to mention where we're starting from is these sorts of protein hydroxylases that recognize these PAPK repeats and introduces several different hydroxylations onto them. And so what we can do, again, using some of the mutations we know about, is to generate a decent starting point for a protein azidase. And so this is something that we're working on now. And I just want to take the last few minutes to talk about enzyme and pathway discovery. We've been working a lot on natural product pathways that produce, non-canonical amino acids, non-canonical bonding to make small peptides, just to ask whether we can develop a new language for making highly modified peptides on the size range of 2 to 5, which is what we see biologically when you don't use something like a non-ribosomal peptide synthase or a RIP. But today, since we're focused on enzymes, I'm going to tell you a little bit about enzyme discovery and one of the challenges for radical halogenases. So these were first discovered by the Walsh group about 15 years ago, starting with CRB2 in basically the synthesis of natural products that are made, that are tethered to an enzyme carrier protein, an acyl carrier protein, or ACP. And so over about 15 years, they've discovered now probably about 6 or 7 different families. Sometimes it's a little bit hard to say where the family starts and stops. But they're all been, serendipitous through studying natural products biosynthesis. And so you can't use sequence analysis to find one family from the next. And this shows you why. For these different families, you can see there's less than 15% homology. So the key motif we're looking for, which is that there's 2 histidines and a missing aspartate around the metal, is very difficult to find using primary sequence methods because it's really confounded by phylogeny and other complex evolutionary events. So what we've done is to use a structural search, again using the AlphaFold database as a way to look for things. And what Yanni has done in the lab is now identified what we think is an atlas of all available radical halogenases of this family. As a preliminary control, he identifies all the seven known families of AKG-dependent halogenases, including ones that are where even though a member had been discovered, they couldn't use sequence space to expand it out further to identify other family members. And so that's initial control. Then we decided to look at just a new family that had an interesting intersection and the types of biosynthetic clusters we found them in. So the first is a little bit boring, but for us was a good control, is he found a new aspartate halogenase. It's the first one for acidic amino acids, but of course, again, it's still in amino acids. I will say that these tend to work on amino acid and peptide substrates. He also found a new class, which we've been very interested in now, that takes biotin and it directly halogenates biotin with this stereochemistry. The reason why we're interested in it is it has an unusually broad substrate scope. It can take all things from dyes, peptoids, fatty acids, aliphatic things, sterile-like compounds. And all that really needs to be there is this carboxylic acid head group, and then it halogenates alpha to that. So for example, this would be good for, if we introduce bromine instead, which this enzyme can do, is to generate non-canonical amino acids from this. And so what Yanni did was solve the crystal structure of BTNX, and what he showed was that the binding site was different compared to our other enzymes, and the fact that it really only made specific interactions in this propionate group, whereas this was more buried in a hydrophobic channel, just making van der Waals interactions. So you can see here, for example, ASP-X, this is a model. BEST-D, we expect there to be a lid that comes over the top of a small substrate. For things like Cure-A, there's no structure of CRB2 bound to its substrate, but we expect the ACP to basically introduce a substrate. But here, we think that this is a good start as an engineerable platform because of the specific binding mode where the R group is mainly bound in a channel. Okay, so with that, I just want to thank everyone who's done the work. I mentioned to you Yanni's work and Eli's work, as well as other people who have left the lab. In terms of the halogenase mechanism, we did some Fro and Marty and Karsten's group. We did XAS with Junko at LBL. And it's been really nice to have Phil and Istvan as people in charge of the NMR and crystallography facilities at Princeton. And thank you, and I'm happy to take any questions.
Thank you. Are there questions? I'll ask a question. So you kind of sliced up these enzymes in order to figure out what was important for this halogenation. You sliced up these enzymes and recombined them. So did you find extended regions that were important, or could you really narrow it down to 4 amino acids, or how small was the sort of secret sauce?
Yeah, because of the, we use a traditional DNA-based approach, so there's a limit to the sizes that you can get. So there was a stretch where where Monica decided to go through and then generate by hand all the different combinations of variants. There's maybe a 6 amino acid stretch where she wasn't sure which needed to come from which sequence. But we did find that a lot of those ended up being on beta strands on one wall of the active site that's involved, we think, in helping signal for closure of the lid when chloride and lysine are bound. Yeah.
Okay.
Great talk. I was wondering, with your new enzyme family that you at the very last described, how did you figure out the substrates of the enzymes? Is it obvious from the clusters that they are located in?
Yeah, so that can be a bit of a challenge. So the ones that are amino acid specific, those ones we can usually tell by just looking at either other amino acid, like a PLP enzyme in the cluster or ATP grasp that forms like dipeptides and tripeptides and so on. And so those, a lot of times we find that will generate some of the modified amino acids first, so then we just screen it against the 20 amino acid panel. For the other one, it was because there's reports in the literature that it was involved in some sort of biotin, the organism had some sort of symbiotic relationship with, it's an algae with a bacteria that involved biotin transport, and it was found in this cluster, and it was one of the few metabolic genes. And so that led to Yanni looking at biotin as a substrate. But otherwise, it can be hard. For example, like he also, in the positive hits that he got, had a hit that we got from screening sequences from primary sequence analysis. Almost all of those are hydroxylases, but we have one hit that we can see because chloride has a +2 isotope, but we don't know what that substrate is. But since we have a hit, we can probably go back and figure out how to isolate it.
How many of those you think you have really hard time to figuring out what the substrates are? Are there a lot of unknowns still, or is a...
Yeah, I think there probably are. Like another one is something that was recently reported in terms of the hits. That's like a polyketide halogenase. Another one is like alkaloid. So they don't just take small molecules and small ACP building blocks. They also take relatively late stage natural product structures. But I think those will be the ones that are interesting to people. Even with WELL-05, people have done a lot of cool applications with it.
All right, one last question.
So you mentioned you use structural similarity to get those cluster of enzymes. So how did you do it specifically? And when you identify these homologs of enzyme based on structure and cluster them into different similarities, were they based on structural similarity or sequence similarity?
No, so what we do is what we did was to take the entire cupin family, which is what it falls under. That's 2 million sequences. It turns out that they're especially have a lot of, so they're thought to be evolved from a his-asp motif that bound to sugar. And so some of them are like just like even transcription factors. Some of them are metalloenzymes, and they go back and forth in evolution from being metalloenzymes and not metalloenzymes. So they're very complicated in terms of primary sequence analysis. And then you have the evolution of all these different families. So you could have like a leucine 4-hydroxylase, and there might be 5 other leucine 4-hydroxylases, and they're not related to each other over the 10% that shows us as a cupin from the fold. And so he basically decided on what the structural constraints were for the specific orientation of where the histidines were, et cetera, and he did a structural search. And that basically, compared to primary sequence search, is also a lot faster, but it still took quite some time to sort through because there's so many sequences.
We do have one more virtual question, and I apologize for my not necessarily knowing how to pronounce this, but they just say, great talk. How about the scalability of Fe2-dependent enzymes? Are the enzyme activities decent to produce a few milligrams of product?
Yes, so actually Merck has been using them quite extensively now. They're fairly well behaved, and we show that we've seen in cell lysate that we can do conversions easily. And we haven't tried much harder than this, like on the over 100 milligram scale. And from what I've been told from scientists at Merck, they feel that they are quite amenable for applications.
Thank you very much. Please join me in thanking Michelle.